Now let's discuss exercise on chapter 1, which is genetic inheritance and genetic control, part B, structured question. Question 2. A. Figure 3 shows a fragment of DNA that is being replicated. So basically for this question, you have to understand the concept of DNA replication. So for question A1, using figure 3 labeled on the diagram, the directionality of the DNA template strand used to synthesize the leading strand. So you have to understand that the leading strand is synthesized from origin of replication towards replication fork. Okay, and then it is synthesized continuously. And in order to synthesize the new leading strand, it has to be synthesized in the direction of 5' to 3'. Okay, so from the diagram you have to determine which one is the leading strand. So obviously the blue strand here with the primer, okay, is the leading strand because it is synthesized from origin of replication towards replication fork. And then the synthesis of the leading strand, it occurs from 5' to 3'. So the question wants you to label the directionality of the DNA template strand used to synthesize the leading strand. So which one is the leading strand? The leading strand is the green strand at the top here. Okay, so this is the strand. This is the template strand used to synthesize the leading strand. So you have to label on the diagram the directionality of the template strand. So the directionality of the template strand would be 5' here and then 3' here. Okay, so this is because earlier, okay, you know that the direction to synthesize new DNA strand is from 5 to 3. So therefore the template, when you write 5 here, so obviously it has to complement with 3. Same goes to this end where you will write 3' here. So obviously on this side is a 5'. Okay, so that is the directionality of the template strand. So be careful when the question wants you to only label the directionality of the DNA template strand, you just label the directionality of the template strand. If you write the directionality of the template strand used to synthesize the Okazaki fragment, here if you label this one, you might not get the mark. Okay, so this is because you are playing safe. Okay, you don't know for certain whether which one is the template strand used to synthesize the leading strand. So you just label one strand that you know for sure it is used to synthesize the leading strand, so that will get you the 1 mark. Okay, so for this next question, you have to circle on the diagram which one is the first Okazaki fragment. So the first Okazaki fragment will be this one. Okay, so the first Okazaki fragment synthesized will be nearer to the origin of replication. Okay, so and Okazaki fragment, they are synthesized away from replication fork. Okay, they are synthesized from replication fork, okay, towards the origin of replication. So why is this the first Okazaki fragment? Why is this not? Why is this fragment is not the first Okazaki fragment? So you have to understand the concept of the DNA replication and the formation of replication bubble. Okay, so imagine the drawing that I've drawn here. The first drawing here is when DNA replication has just started. Okay, so when DNA replication has just started, the template strand, okay, will separate and becomes the template, and this will start the formation of the replication bubble. Okay, so the replication bubble at the start of DNA replication is only small. Okay, so this will synthesize the first Okazaki fragments. So as replication of DNA occurs further, okay, and the replication bubbles open up even further, so this will synthesize the next Okazaki fragment, which is fragment 2. And as replication DNA proceeds further, okay, and open up the replication bubble even bigger, okay, so this will synthesize the next Okazaki fragments. So these are the Okazaki fragments synthesized for the lagging strand. Okay, so as for the leading strand, as you can see, they are synthesized continuously as the replication bubbles gets bigger and bigger. So the leading strands are synthesized continuously from 5' to 3' end. Okazaki fragments are also synthesized in the same direction, which is 5 to 3'. Okay. Question 3. It was found out that the DNA polymerase 1 in the eukaryotic cells undergoing DNA replication was non-functional. How would that affect the synthesis of the leading strand? So, as you know, the function of DNA polymerase 1 is to replace RNA nucleotides of RNA primer and replace it with DNA nucleotides. So if DNA polymerase 1 is not functional, so therefore the RNA nucleotides of RNA primer will not be removed and replaced with DNA nucleotides. So that is for 1 mark. Next question. Several human genetic disorders are associated with the alterations in the chromosome number or structure. So complete the table below. So for the first genetic disorder, which is chronic myelogenous leukemia, which involves the exchange of a large portion of chromosome 22 with a small fragment from a tip of chromosome 9 producing a Philadelphia chromosome. So basically this type of mutation is reciprocal translocation, okay, that involves the alteration of chromosome structure. Okay, reciprocal translocation that involves alteration of chromosome structure. For the next question, which is cry to chat, the description of this disease is that it is due to the specific deletion in chromosome 5. So therefore, this involves deletion. Okay, when a segment of a chromosome is deleted, so this means that it involves the alteration of chromosome structure as well. Okay, so cry to chat is deletion that alters the chromosome structure. For the third genetic disorder, which is Down syndrome, that involve trisomic for chromosome 21. So that means chromosome 21 has 3 chromosomes. So this the type of mutation involved is trisomy 21 that involves the alteration of chromosome number. So 1 mark for each correct answer. Question C. Figure 4 shows the lac operon in E. coli. So for this question, you have to label all components of the lac operon correctly for 1 mark. Okay. So when you label all the component of the lac operon correctly, you will get 1 mark. So the first region here is the regulatory gene. Okay. And then you have the promoter, and then the operator, the LAC-Z gene, the LAC-Y gene, and also the LAC-A gene. So you label the segment correctly, then you get 1 mark. So just remember, for the regulatory gene, it is to produce, or it codes for the synthesis of repressor protein. Promoter is the site where RNA polymerase will bind to. Operator is the site where repressor will bind to in the absence of lactose. LAC-Z gene is to produce the enzyme beta-galactosidase. Likewise, gene is to produce the enzyme lactose permease, and LAC-A is to produce transacetylase enzyme. Okay. Question 3. Explain the effects of gene expression of the like operon in the presence of lactose. For the like operon, you have to understand the two situations to explain the function of like operon. So the first situation that you have to understand is what happened to the like operon in the presence of lactose, and also what happened to the like operon in the absence of lactose. So for this question, you have to explain what happened to the gene expression when lactose is present. Okay, so lactose is present, it means that the lactose are present in the bacterial cell. The bacterial cell has taken up lactose from the medium that it is grown in, okay, or the medium or the culture that it is grown in. So this question it worth 3 marks. Okay, so earlier we have looked at the component of the like operon that consists of the regulatory genes, the promoter, the operator, the structural genes, which is the like Z, like Y, and the like A. So each of the region has its own function. So for the regulatory gene, it is to synthesize repressor protein. So repressor protein that has been synthesized will bind to the operator and prevents the transcription of the mRNA for the 3 genes. Okay, that is in the absence of lactose. In the presence of the lactose, when lactose enters into the bacterial cell, lactose will be converted into its isomer, which is allolactose. So allolactose will bind to the repressor protein that was produced based on the information from the regulatory gene. So once the allolactose bind to the repressor protein, it causes the repressor protein to change shape. Okay, and once the repressor protein changes its shape, it cannot bind to the operator. Okay, so now the operator is free, it is not blocked. So the operator is activated or switched on. So once the operator is free, then the RNA polymerase, okay, can bind to the promoter. So promoter is the site where RNA polymerase will bind to, and then the function of this RNA polymerase is to transcribe or synthesize mRNA, okay, a long single mRNA for the 3 structural genes. Okay, so that is in the presence of lactose. So once a single mRNA has been synthesized, so the mRNA is used during translation to produce 3 different enzymes. Okay, so for the region that copies for the region on the mRNA that copies the information from like Z gene, it produces the beta galactosidase enzyme. Okay, so the function of this enzyme is to hydrolyze lactose into glucose and galactose. As for the like Y gene, it is to synthesize the enzyme lactose permease, okay, that allows lactose to enter into the bacterial cell. And for the like A gene is to synthesize the enzyme transacetylase. Okay, so that is in the presence of lactose. So for the 3 marks, you can write all the sequence of event that occurs any of these events, okay, for 3 marks. Okay, so which I've already explained. Number 1, lactose enters into the E. coli and converted into allolactose. So allolactose then bind to the repressor protein. Repressor protein then changes its shape and causes it to cannot bind to the operator. So the operator so therefore now the operon is activated or switched on. The like operon is switched on. So next is RNA polymerase can bind to the promoter. Transcription of structural gene or like Z, like Y, and like A genes can be used to synthesize a single mRNA. Okay, so from that mRNA, translation will occur to produce the 3 enzymes, which is beta galactosidase, lactose permease, and also transacetylase. And then from the 3 enzymes, then lactose can be hydrolyzed using the enzyme beta-galactosidase, and the lactose is hydrolyzed into glucose and galactose. Okay, so basically the purpose of hydrolyzing lactose into glucose and galactose is so that the bacterial cell can use this monomer, the glucose for example, to undergo cellular respiration to generate energy. So that is for the 3 marks. You can choose any of these points.